This protocol demonstrated a simple and producible method to assess phagocytosis ability of macrophages using EGFP-expressing Escherichia coli.
Hello, my name is Zhang from the Second Hospital of Dalian Medical University. Today, we are going to show you an easy and visualized way to assess macrophage phagocytosis, which can be easily done within 2 hours. This method was widely used in the study of the innate immune function. It is believed that the innate immune function remains intact in the elderly. So today we are going to isolate the peritoneal macrophages from the aged and young mice and to see the phagocytic ability of these two groups. Well, let's get started.
Firstly, synthesize the EGFP gene fragment and clone the fragment using high-fidelity Taq DNA polymerase. Then, ligate the PCR product into the PET sumo vector. Thirdly, transform the ligation products into the E. coli strain and induce EGFP expressing with lactose. These EGFP expressing E. coli served as the marker for the phagocytosis assay. Next, the mouse peritoneal macrophages were isolated and cultured. Then, EGFP expressing E. coli were co-incubated with the macrophages for 1 hour at 37 degrees centigrade. After the quenching step, the macrophages were ready for assessment by both fluorescence microscope and flow cytometer. Synthesize the EGFP gene fragment and amplify the fragment with the forward and reverse primers using high-fidelity Taq DNA polymerase to ensure that the PCR products had single 3-end adenine.